course, after we look at cell structure and function, we'll go on and look at transport system itself. Transport systems in the cells. And after that, we'll look at macromolecules. Macromolecules. Another literature or other books, you may find that it is called biological molecules. What is that? Biological molecules. What do we look at under macromolecules or biological molecules? Under this one, we're going to look at amino acids and proteins. Then we'll look at carbohydrates. Apart from carbohydrates, we also look at nucleic acids. Nucleic acids. That is the DNA, the nucleic acid, and the RNA. Thereafter, we'll look at lipids. Lipids. After lipids, we can also consider looking at water. And now we'll look at both the physical as well as chemical characteristics of water. Physical and chemical characteristics of water. After unit 3, we go to unit 4 where we look at photosynthesis. Photosynthesis. Under photosynthesis, we look at the concept of the two stages of photosynthesis. And consider looking at the first one, the light-dependent reactions. Light-dependent reactions that are occurring in the thyroid membranes. That are occurring in the thyroid membranes. Secondly, we look at the Calvin cycle. The Calvin cycle also refers to as the dark reaction. The dark reaction. Thereafter, we'll conclude by looking at Mendelian genetics. Mendelian genetics. If there will be any additions, we'll be able to communicate as we move on to this particular course. Okay. So this is a tentative course outline, and these are some of the key concepts that we are going to look at in this particular course. So to begin with, we begin with unit 1, which we are calling cell structure and function. Cell structure and function. Cell structure and function. So what is a cell? Anyone who can help me define what a cell is? What do you understand by a cell? What is a cell? Yes. So a cell is the smallest unit of a living thing or a living or an organism, so to say. Also. So a cell is defined as a basic unit of a living organism. Are we together? That is a cell. I'll give you a practical example so that you can be able to understand. Look at this building, nice huge campus, right? They say about 4 or 5 story building also. How did it come to be? What did engineers use to build this building? What were they using? It's a block. So they started with the foundation, 1 block, 2nd block, 3rd block, and it came to be a 5-story building. So in this case, to liken it to a cell, we are saying the block is a basic part of this building. So it is actually a block, 1 block, together with other blocks, put together, they made what is the ICU campus now altogether. So that block is very important also. Without it, they wouldn't have this building altogether. So that is how important a cell is. That is important. That is how important a cell is. It's a basic unit of a living organism altogether. So now, you need to understand that a group of cells, okay, with the similar functions or similar characteristics, working towards a particular function, they form what is known as what? What do they form? A group of cells working together, similar, not system, a tissue, exactly. So a group of cells with similar functions, with similar characteristics, of course, they form what we call a tissue. A group of tissues working together, what do they form? Tissue system, also. A tissue system. A group of tissue systems working together, what do they form? This system that we are talking about, for example, we can say respiratory system, we can also say respiratory system, so some examples that we can give. Respiratory system. So a group of these systems working together, what do they form? Exactly, they form what is known as an organ. Okay? Example of an organ is what? The heart, for example. What else? The liver, the brain, etc., and so on and so forth. So these are organs. So now, a group of organs working together in a system, what do they form? The body. An organism. They form what? An organism. So the point that we are trying to say is that everything is emanating from what? A cell. So a cell is a basic unit of a living organism. So as you see, from cell we have tissue system, organ, then we have eventually an organism. And we have got a lot of organisms that exist also, including human beings. We are organs because we respire also. We are living things altogether. So that's how important the cells are. So now, one thing that we need to understand is that there are millions and billions of cells that exist also. We can give examples: red blood cells, white blood cells, and so on and so forth. Even stem cells and so on and so forth. So there are quite millions of cells that exist, both in plants as well as in human bodies or in human beings. There are billions of cells. But one thing that we need to note is that these cells, they perform specific functions. And they have different functions as well as characteristics altogether. So now, there is a theory which we call the cell theory. You can write it down. Cell theory. Cell theory. What does the cell theory state? What does the cell theory state? So the cell theory states that a cell is a basic unit of a living organism. That is the first statement. A cell is a basic unit of an organism, and that all organisms are made up of cells, and that all organisms are made up of cells. The last statement in the cell theory states that all cells come from the pre-existing cells altogether. So how many statements have I made in the cell theory? 3. Number 1, that a cell is a basic unit of a living thing or an organism. which we have already talked about. And secondly, we said that all organisms are made up of a cell, which is true. And then lastly, we said all cells emanate or come from pre-existing cells. 3 statements, that's what a cell theory states. Okay. So now, pre-existing cells. So to make you understand that pre-existing cells, what it means is, I would ask a question to this madam here. Where did you come from? Yourself, where did you come from? Where did you come from? How did you come into existence? How did you come into existence? Is it not from your mother and your father? Not so. So those are already pre-existing ones. Or human beings, right? Then your mother, where did she come from? From your mother, father. Then your father also came from there. Just like that. This is the principle. Have you ever heard of mitosis? Mitosis is the division of cells, not so. One cell will divide into 2, and then that other cell will divide, exactly mitosis. So all cells come from already existing cells through the process of mitosis. Okay. So that's what that statement means. Now, we need to also understand how are these cells studied. The question is, how are these cells studied? Scientists, biologists, agronomists, how do they study these cells? Of course, they use what is known as a microscope. Have you ever heard of a microscope? So a microscope can be defined as an instrument that is used to magnify objects, tiny objects that we are able to see with our naked eyes. Altogether, the microscope is defined as an instrument in the laboratory that is used to magnify tiny objects that we are able to see with our naked eyes. The most prominent microscopes, there are 2. There's one that we call electron microscope and the light microscope. Okay? Now, we need to understand what is the difference between the 2 microscopes. An electron microscope is an instrument that uses a beam of electrons in order to magnify the specimen. It uses the beam of electrons. And then a light microscope uses light in order to magnify the specimen. So that is the major difference, that they will all give you the same result. They will all give you the same result. So microscopes are studied through, cells are studied through the microscopes. So to say that. So now, let's continue looking at the cells. So now, when scientists did a lot of research, a lot of experiments, and a lot of studies on cells, they realized that within the cells, remember I mentioned that there are billions of cells also. So within the cells, there are those cells that share common characteristics. There are those cells that have similar functions. So what they did is that they categorized these cells, or the group of cells, into 2 broad categories. Number 1, they called them eukaryotic cells. Then the second category is known as prokaryotic cells. So there is eukaryotic and prokaryotic cells. I repeat. So the criteria that they were using in order to categorize these types of cells is they were looking at first the characteristics, the phenotypic appearance of the cell. When viewed under a microscope, what are the physical characteristics, the phenotypic, what we are able to see. Then also, they looked at the cell components. Cell components, what are some of the characteristics, what are some of the components that they have that the other cells or group of cells don't have. So based on those recommendations, based on those discoveries and studies, they came up with those 2 categories. One, we are talking about eukaryotic and then prokaryotic cells. So we'll start looking at prokaryotic cells first. What is prokaryotic cell? What are prokaryotic cells? Everyone with an idea? What are prokaryotic cells? Let me define what prokaryotic cells are. Prokaryotic cells. What are these cells? Prokaryotic cells. Everyone with an idea? We are talking about the two categories, eukaryotic as well as prokaryotic cells. What are these prokaryotic cells? How do we define what prokaryotic cells are? Anyone? Then we write this down. So a prokaryotic cell is a single unicellular cell. A prokaryotic cell is a single unicellular cell that lacks a nucleus and the other cellular bound organelles. When I use the term organelles, what I mean is cell component. Cell component, cell organelle, that is the same language. So I'll repeat. The prokaryotic cell is a single unicellular cell that lacks a nucleus as well as other membrane-bound organelles. What are some of the examples of prokaryotic cells? We have the bacterial cell, okay? Bacterial cell, and then we have, that is the first one, and then the second one we have the algae. These are small microbes, okay? So they are in the same category as bacterial cells. So these are examples of the prokaryotic cells. And that, I repeat, these were categorized based on their typical characteristics. So now, quickly, let us do a sketch of these prokaryotic cell. So one thing that you need to understand is that in terms of complexity, prokaryotic cells are more complex than, eukaryotic cells are more complex than prokaryotic cells. Prokaryotic cells are not so complex. They are not so complex. So what are some of the organelles that this prokaryotic cell has? We'll start with the plasma membrane. Okay. Write down plasma membrane. The plasma membrane. What is a plasma membrane? We need to understand what a plasma membrane is. A plasma membrane is an outer covering of the cell. The plasma membrane is an outer covering of the cell that is selectively permeable. What do I mean by selectively permeable? Meaning, it is able to allow certain substances in the cell, in the cytoplasm, and the other substances not to go in the cytoplasm. So it is selective. Other substances will be allowed to enter into the cell from the extracellular fluid, and other substances won't be allowed to enter the cytoplasm. And vice versa. Toxic substances will be chucked out of the cell, through the plasma membrane, and using substances will be maintained within the cell. So it is selectively permeable. And one thing that we need to understand is that this plasma membrane is made up of a phospholipid bilayer. I will write the spelling here in case you are wondering how to write a phospholipid bilayer. So, what is a phospholipid bilayer? What is a phospholipid bilayer? You can write down a phospholipid bilayer is a lipid molecule with that is composed of two fatty acid chains. So I'm saying a phospholipid bilayer. By the way, bi means two, so meaning it has a double layer. So it is a lipid molecule. A phospholipid bilayer is a lipid molecule that consists of, number one, two fatty acid chains. Number two, it consists of a glycerol backbone. And lastly, which is the third one, it consists of a phosphate group. So I've mentioned three things. So this phospholipid bilayer is the one that actually makes up a plasma membrane. Or in other words, we can say a plasma membrane is made up of a phospholipid bilayer, which we have defined. Are we together? So that is about the plasma membrane. Outer covering, it also protects the interior of the cell from the exterior. Are we following? Selectively permeable, allows in and out of the cell elusive substances, and deters elusive substances from leaving the cell and toxic substances from entering into the cell. Are we following? So basically that is that. Then we move on to what we call the cytoplasm. Who has an idea what the cytoplasm is? Who has an idea what the cytoplasm? You guys, this is grade 2 of biology. Cells. You never did. It has been long since you left high school. Because we used to learn these things. A long time. Cytoplasm, who has an idea? So basically the cytoplasm is a gel-like fluid that is found inside of a cell. In this case, we are talking about, or specifically, we are talking about the prokaryotic cell. To be precise, a bacterial cell. So this fluid contains about, or rather its composition is about 70 to 80% is water. How together? 70 to 80% of its composition is made up of water. What about the 20%? The 20% is what we refer to as a semi-solid. The 20% is what we refer to as a semi-solid. And in this semi-solid, what we have is different types of substances, ranging from proteins, amino acids, ions, carbohydrates, which is sugars, polysaccharides, and so on and so forth. That is the semi-solid part of the cytoplasm, because we are saying about 70 to 80% is water. Now, 20% is the semi-solid, which is composed of different types of substances, which I've actually tried to mention. Sugars, which is carbohydrates. Under carbohydrates, you know that we have monosaccharides, oligosaccharides, disaccharides, and so on and so forth. Then we have ions that are found in the cytoplasm, like sodium, potassium, and so on and so forth. Okay, nucleic acids, DNA, RNA, those specifically, the DNA in the prokaryotic cell is housed in the nucleon, which we are going to talk about very soon. How to get? So now, we need to understand the core function of this cytoplasm within the cell. What is the core function of the cytoplasm within the cell? The most importantly is that this cytoplasm within the cell, this is where all the organelles, cells, organelles, or rather other cell structures or components are housed. So they are suspended in the cytoplasm. Other cell organelles, other cell components are actually housed within the cytoplasm. How we follow? Other cell components or organelles are housed within the cytoplasm. So it's very important also. Number two is that the other function, the second function of the cytoplasm is that almost all the metabolic biochemical reaction within the cell occurs within the cytoplasm. All cells metabolic as well as biochemical reactions occurs within the cytoplasm. How do they? It occurs within the cytoplasm. I hope you have picked those two functions that I mentioned. Two important functions. This is where all organelles are suspended. Secondly is that this is where all biochemical as well as metabolic reactions in the cell occurs. How to get it? Again, if you look closely, you find that usually these bacterial cells, they have tail-like structures. How to get it? Tail-like structures. And these tail-like structures are referred to as the flagella. That is the plural, and then flagella is singular. So the flagellum is actually a tail-like structure, and when present usually it is in one or two. Usually there are not many present, as opposed to the cilia. The cilia, usually the lining of the intestines, when present, they are usually many in number. The cilia. But when you look at the flagella, usually the tail-like structure of present, say that it is one or two to the max. How would you get it? The sperm cell has got a flagella, not so. So now, what is the major function? What is the major function of this flagella to a prokaryotic cell? Why is it there? What is the major function? Yes. Exactly. So the major function of this flagella is for locomotion or propulsion. Are we together? For locomotion or propulsion, the cell movement, easy cell movement. Are we together? So that function is very simple to understand. It is very simple to remember. And also, locomotion or propulsion, so that it helps the cell to move around. Okay. What other function or cell organelle that we know? You've seen these hair-like structures, right? That I was drawing. These are referred to as the pili. The pili. So now, these pili are hair-like structures, and they have got a specific function, and that specific function is in reproduction. And in a special kind of reproduction referred to as conjugation. I'll write it down. Referred to as conjugation. Conjugation. Okay? In a special type of reproduction known as conjugation. So basically the pill, or rather the pill's function is for reproduction. What else can we talk about? Have you seen the dark region? This is the dark region in the center of the cell. The dark region in the center of the cell is referred to as the nucleoid region. What is the function of the nucleoid region and how is it defined? So the nucleoid region is a region in the center of the prokaryotic cell. It's a dark region in the center of prokaryotic cell because it is holding at the center of the prokaryotic cell. Now the question comes in, what is the function of the nucleoid region? It must be mentioned that in prokaryotic cells, the DNA is housed in the nucleoid region. You will see when you start looking at eukaryotic cells like the plant cell, animal cell, you will discover that there's a difference where the DNA is housed in the eukaryotic cell or in the plant cell and in the prokaryotic cell. In the eukaryotic cell and in the eukaryotic cell, you will see that it is housed where? In the nucleus. And from the definition here, for prokaryotic cells, we have mentioned that the prokaryotic cell does not have a nucleus. It lacks a nucleus. That is the key thing or the key concept. It lacks what? A nucleus. And therefore, and that is the reason that the DNA is housed where? In the nucleoid region. In the nucleoid region. I hope we are following. What else have we not talked about? We have also what we call the cell wall. Cell wall. What is a cell wall? Everyone with an idea? What is a cell wall made up of? And what is the function of a cell wall? Exactly. So number one, the cell protects the interior of the cell. Just like the plasma membrane, it protects the interior of the cell from the extracellular environment, or rather the outside environment of the cell. Okay, that is number one. Number two, the cell wall maintains the shape of the cell. The cell wall maintains the shape of the cell. Number three, who can tell me number three? What is the other function of the cell? Very important. Yes.
It prevents the
Exactly. So the cell wall prevents dehydration. For those of you who are wondering what dehydration is, we are talking about for dehydration, we are talking about the loss of water from the cell, not the loss of water from the cell. That's what we are talking about. So it prevents dehydration on top of protecting the interior of the cell from the outside environment, extracellular fluid, and also from maintaining the cell's shape. It is actually very important. It's actually very important. What else have we not talked about? Then also we have what we call the capsule. In between the plasma membrane and the cell, there's what we call the capsule. Okay? Capsule. So this capsule is very important because it gives the cell's ability to attach to its surfaces in the outside environment. The function of the capsule is to give the cells the ability to attach itself to other cytoplasmic environment outside of the cell, or in the extracellular fluid, or in the outside environment of the cell. Is that clear? That is the function of the capsule. Very important. Okay, so what else have we not talked about? I think we have exhausted all the cell organelles in a bacterial cell, unless we have a question. Oh, sorry. Just what is the function of the ribosome? So the functions of the ribosome is simple. The function of the ribosomes is to build up the protein synthesis. How to get it? So it builds up the proteins. How to get it? From amino acids to translate them into proteins. So it builds up the proteins. So it's for protein synthesis. How to get it? Any questions about the prokaryotic cells before we move to the next? Yes.
In the
Ribosome.
yes,
So the function of the ribosome is for protein synthesis. And when we talk about synthesis, it's building up of proteins. So it is involved in the process of building up of proteins from their raw form, which is amino acids, to proteins. So it plays an important role in that aspect. Any other question? Any other question? So if we don't have any other question, then we move to eukaryotic cells. We move to eukaryotic cells. Remember, we mentioned that there are two types of cells. What we are from talking about, or rather what we are from discussing, is the prokaryotic cells. And like I mentioned, in terms of size, complexity, the eukaryotic cells are more complex. And in terms of size, the eukaryotic cells are larger than the prokaryotic cells. In terms of organelles, the prokaryotic cells has fewer organelles as compared to the eukaryotic cells. The eukaryotic cells have got fewer components or cell organelles as opposed to the eukaryotic cells. Now, what is the size of prokaryotic cells? So, prokaryotic cells ranges from, so right, cell size. Prokaryotic cells ranges from 0.05 micrometer. The SI unit is M, and then like this, okay, micrometer.
So
0.05, that is prokaryotic. 0.5 to somewhere 5 micrometer altogether. Then the eukaryotic is from 5 to 100 micrometer altogether. That is the cell size. So even in terms of size, you find that eukaryotic cells are larger than prokaryotic cells. In terms of complexity, because it's larger and definitely it has more organelles than prokaryotic cell has. Okay? So let us define what eukaryotic cells are. Eukaryotic cells. Eukaryotic cells. What are these cells? This type of cells? Eukaryotic cells. Who can define what an eukaryotic cell is? Yes, try to define it.
Is a cell that has a membrane bound and other membrane bound compatible cells.
Exactly. So now, when we talk about eukaryotic cells, we are looking at a simple single unicellular cell that has a nucleus and the other membrane bound organelles. Are we together? So that is the difference between prokaryotic cell and the eukaryotic cell. So in this case, we have a cell that has a nucleus. But when we are talking about prokaryotic cells, if you recall, we said prokaryotic cells lacks the membrane-bound nucleus. But with eukaryotic cells, it has a membrane-bound nucleus plus other membrane compartments or sacs or rather organelles. Is that clear? Now, what are some of the examples of eukaryotic cells? Let me write them down. Number one, we have what? Animal cells, or rather plant cells. Number two, we have animal cells. In the third, we can give examples of protists. Four, we can also talk about the fungi. Okay, so those are the examples of eukaryotic cells. Those are the examples of eukaryotic cells. But now, you need to understand that by the end of this course, or rather by the end of this topic, you should be able to differentiate between a plant cell, that is the objective of this topic. You should be able to differentiate between a plant cell and an animal cell, and between an animal cell and a plant cell, and between a plant cell and a prokaryotic cell, and between an animal cell and a prokaryotic cell. So you need to understand what are some of the cell organelles or other cell components that are found in a plant cell, they are not found in an animal cell. What are organelles that are found in a plant cell and they are not found in a bacterial cell or the prokaryotic cell? What are some of the organelles that are found in a bacterial cell that are not found either in a plant or an animal cell? So those are the objectives, and you need to also ensure that you know the specific functions of each organelle, or rather of each cell component or structure. Are we together? So quickly, we look at the plant cell, for example. We start looking at the plant cell. So what I'm going to sketch here is a typical illustration of a typical diagrammatic illustration of a plant cell. A typical diagrammatic illustration of a plant cell. What we are looking at was a typical diagrammatic illustration of a bacterial cell, a prokaryotic cell. So what we have here, of course, like we said, usually the plant cells are somewhat complex than the bacterial cell. So what we have here, let me start with the nucleus itself. We'll try and sketch the nucleus itself. Okay. And here. sketched here, what I've sketched here is what we call the nucleus. How do you get it? The nucleus. Remember that the eukaryotic cells have got what? A nucleus. And what are we saying? Is that this nucleus has got other components. How do you get it? It has got other components. What are those components? So it's a nucleus as a whole. It's called a nucleus, but it has got other components. What are those components that we are talking about? Firstly, it has got what we are calling a nuclear envelope. Nuclear envelope. The nuclear envelope. What is an envelope? Outer covering, not? Even when you are sending it, they put it in the envelope. It's something that is covering the inside, the interior of your whatever that you have put in there. So even in this case, the nuclear envelope protects the interior of the nucleus. Now, you need to understand that this nuclear envelope is a double membrane structure. It's a double membrane structure. As you can see the way I've drawn it or sketched it, there is it. So I'm going to say this nuclear envelope is a double membrane structure. So we have got the inner membrane, and then we have got the outer membrane. Inner and the outer membrane. Then as you can see here, there are these small holes that I have drawn or that I have sketched. These are called the micropores. How do they? Micropores. They are small holes known as the micropores. Micropores. Okay? These are some of the pores that allow substances in and out of the cell, especially the mRNA. mRNA, when it undergoes what we call transcription, changes from DNA to RNA transcription, and it goes into now the rough endoplasmic reticulum, okay, for protein synthesis. Okay. So these small holes basically is to allow substances such as the mRNA that has undergone transcription from the DNA to go into the rough endoplasmic reticulum. Okay. So basically, therefore, substances movement in and out of the in and out of the nucleus. What else do we have? So micropores. Then we also have what we call we have what we call the nucleolus. Also, nucleolus. What is the function of the nucleolus? What is the function of the nucleolus? Yes. Exactly. This is the place where ribosomes are formed. Exactly. This is the place where ribosomes are formed. And actually also ribosomes, they actually also diffuse through the pores and goes through the rough endoplasmic reticulum. What else have we left here? We have talked about the nuclear envelope, the nucleolus, and so on and so forth. Oh yes, we left out there the DNA, which is known as the chromatin. Okay? Chromatin, how do you spell chromatin? That is C-H-R, chromatin. Yes, chromatin. What is this chromatin? So chromatin is referred to as chromatin because it has got DNA. So chromatin is actually DNA, deoxyribonucleic acid plus associated proteins plus associated proteins, but these proteins are called the histones. Okay. To be specific, the proteins that we are talking about are histones. So chromatin is DNA plus the histones, okay, plus the histones. And we cannot also go without talking about the same ribosome, okay, the same ribosomes. I think I mentioned earlier on the function of the ribosome. I said the function of the ribosome is actually to form, or rather to synthesize the protein. So it's protein synthesis. That is the core function of the ribosomes. So that is what the nucleus in the eukaryotic cells is actually made up of. That is the nucleus. Are we together? So now, you need to understand, or rather it must be mentioned that in eukaryotic cells, the DNA plus associated proteins plus histone is housed where? In the nucleus. However, in the prokaryotic cells, it is housed in the center dark matter that is known as the nucleus. So that is the difference between where the DNA is housed in prokaryotic and the eukaryotic cells. Do you have any questions? Or we wrap up for today, we can end here. We are going to continue from here and then meet tomorrow. Have a blessed day. Thank you very much.